Okay, today I'm going to talk to you about genetic engineering. So genetic engineering is all about modifying an organism's genome to introduce desirable characteristics. So the example we're going to look at today is being able to produce bacteria that can make insulin. And insulin is used for people with diabetes. Okay, so the first thing you need to do is you need to remove the desired gene. So within an organism's genome, there will be the gene for insulin. So this insulin, remember, is a hormone which basically helps to regulate blood glucose levels. So the first thing you need to do is remove the desired gene. So we need to kind of cut that gene out. And in order to do that, we use something called a restriction enzyme. Now this restriction enzyme cuts the DNA at a very particular part in order to remove that insulin gene. Now, when I think of restriction enzymes, I think they're a little bit like scissors because they're used to cut or chemically cut the DNA in a particular sequence. Now, what's left are these things called sticky ends. Those are the bits on the end of that bit of DNA, that particular gene, which we can then, well, we'll use it in a little while, and I'll show you that. So at the moment, all we've done is isolated that insulin gene by using a restriction enzyme. We've taken out the gene we want. This is the insulin gene. That would produce insulin. Okay, next thing that we need to do is we need to remove a plasmid from a bacterium. And we'll see why in a minute. So, remember, bacteria are microorganisms, small living things. Now, their structure is different from our cells. So, a lot of bacteria have this thing called a plasmid. A plasmid is a real simple, just circular bit of DNA that's quite simple, and that's why we use it. We use it in genetic engineering as a vector. Now, all a vector is something that carries something else. And we're going to see what that is in a minute. So we remove this, again using enzymes, we remove the plasmid, and here's the plasmid right here. Now, what we do is we use the same restriction enzyme from before. So we use this in order to cut a section out of this bacterium. Okay? Again, we've created some sticky ends. Right, now, the next step is what we're wanting to do is we want to get this insulin, the gene that we want, and put it into our plasmid. We'll find out why in a minute. So number 3 is to put the gene into the vector. Okay, so in this case, the vector is a plasmid, the thing that's going to carry our DNA, our desired DNA. So here's my plasmid. Remember, it's had this section cut out using restriction enzymes. And here is my desired gene, insulin. So I want to put that into the plasmid so it's incorporated in the plasmid's DNA. To do that, I use another enzyme, which is called ligase. Now, whenever I think of ligase, I think it's a little bit like glue. It's a little bit like glue because it helps to stick this bit of DNA, our desired gene, into the plasmid. So ligase is a little bit like glue. So now we have the DNA within the plasmid and the DNA from this gene here, the insulin gene. It is now called recombinant DNA. That just means we've now combined the DNA from our desired gene with the vector, in this case a plasmid. So number 4, the next step, is we need to put the plasmid or the vector. Remember, plasmid is the example of a vector in this case. So plasmid is inserted into bacterium. Okay, so Now, here is a bacterium. Oh. So what we're doing now is we're taking that recombinant DNA, so the plasmid with our desired gene, and we're putting it back into that bacterium. So here it is, it's now within the bacterium, so it's still got the plasmid is there, but now it contains the gene for insulin as well. We've put it in there using ligase, incorporated it into the DNA of the plasmid. So the next step, and the final step, is we allow the bacteria to reproduce. So the bacteria reproduce and make insulin, which we can harvest. So all that we do is, all of these bacteria, they're very good at dividing, making copies of themselves. They reproduce asexually to make lots and lots of copies of themselves. And as they grow and as they make copies of themselves, they also copy that gene for insulin. So as they're reproducing, the whole time they're producing insulin, which can then be harvested and we can use to help people with diabetes. So we've harvested all that insulin. Now that is how we can genetically engineer using plasmid as a vector. It's just worth saying that viruses can also be used as vectors. So because viruses insert DNA into organisms they infect, they could also be used to introduce the DNA into a target organism. So that is how genetic engineering works. Thank you.